We will be discussing the various cell organelles and their function within the cell, and in this we are going to take up the lysosomes, which are of course called as suicide bats. It was discovered by Christian Didouet in 1955 in the liver cells and called it as pericanalicular dense bodies. Nevikov called it lysosomes, and he also gave the term GERL, that is, relation between Golgi body, endoplasmic reticulum, and lysosome is in short known as GERL system. Lysosomes are abundantly found in fungi. Pitt and Galvin, 1973, confirmed their presence in the higher plant cells. That is, he observed it in potato stem. With the exception of mammalian RBC, they are reported from all animal cells. Here you can see lysosomes are spherical bag-like structures covered by a single unit membrane, and lysosomes are polymorphic cell organelle. That is, you will study primary, secondary, residual bodies, and autophagosome. Primary lysosomes are protolysosomes. They are vesicles of Golgi body which contain hydrolytic enzymes or digestive enzymes are known as primary lysosomes, and these primary lysosomes stores acid hydrolases in inactive form, and they contain approximately about 40 different types of digestive enzymes. Proteases, that is protein digesting enzymes, galactosidase enzymes connected with carbohydrate digesting metabolism, amylase, beta galactosidase enzyme, etc. Phosphatase group, say for example ATPase, nuclease group, nucleus digesting enzyme, that is RNAase and DNAase, esterases group, for example phospholipase enzyme. Lysosomal membrane contains stabilizers. Substances which stabilize lysosome membrane stop its rupture, thus prevents autolysis. Say for example cortisone, cholesterol, chloroquine, etc. Labilizers is also present in lysosomal membrane, and these are referred as destabilizers. Secondary lysosome or digestive vacuole or heterophagosome. You know, when the primary lysosome fuses with phagosome, it results in the formation of secondary lysosome or heterophagosome. Tertiary lysosome or residual bodies are Telolysosomes. Lysosome containing undigested material. They are eliminated by exocytosis, but sometimes they remain inside the body and they are responsible for causing certain diseases like congested heart failure, hypertension, etc. Autophagic lysosome or cytolysosomes or autophagosomes. Lysosome, sometimes they digest the old dead or old cell organelles, and this is called as autophagosomes. Here you can see the primary lysosomes fusing with the phagosome to form heterophagosome or secondary lysosome. Then the digestion taking place, digested material pass through the matrix, resulting in residual bodies, and these residual bodies are excreted out by exocytosis. Intracellular digestion, it is of two types. Heterophagy, digestion of foreign materials received in cell by phagocytosis or pinocytosis. Secondary lysosome performs as the intracellular lysosomes. B. Autophagy, digestion of old or worn out cell organelles. Autophagy also takes place during the starvation of the cell. Here you can see the autophagy, that is digestion of the intracellular organelles. Ambylysosome, lysosome which perform both heterophagy as well as autophagy. Extracellular digestion. Lysosomes of osteoclasts, that is bone-eating cells, dissolve unwanted part of bones. Extracellular digestion by lysosomes also taking place in fungi. Climinate. Conversion of complex and inactive hormones into simple and active hormone through lysosome is called climinophagy. Say, for example, thyroglobulin, lysosome converted it into thyroxine. Thyroglobulin is complex and inactive, resulting in the formation of simple and active thyroxine. Cellular digestion, that is autolysis. Digestion of whole cell through lysosomal enzyme. Sometimes all lysosomes of a cell burst to dissolve the cell completely. Old cells are removed by autolysis. Unwanted organs of embryo are destroyed by autolysis. Catespin of lysosome digests the tail of the tadpole during metamorphosis. Untimely rupture of lysosome sometimes results in mutation. You can see the autolysis.